So we are witnessing a quiet revolution in brain science, shifting from traditional diagnosis to detailed cell state maps, from reactive treatments to proactive or preventive cares, and also from one-size-fits-all approaches to mechanism-matched care. So in this panel discussion, we will explore the convergence of single-cell and spatial omics, biomarkers, AI, and regenerative medicine. So these groundbreaking discoveries are moving from the lab to the clinical practice. So together we'll discuss how we can integrate them into clinical practice and serve society. So first, I would like to introduce our three distinguished panelists. Very short intro. So Dr. Giulio D'Angeli is the co-founder and CEO of Oxbridge Clinical, a pioneering diagnostic company developing rapid point-of-care technologies that utilize various sample types, including blood. And he is dedicated to transforming complex biological data into fast, scalable diagnostic tools that can influence clinical decisions. And his work aims to enhance patient outcomes through innovative diagnostic solutions. Our second panelist is Dr. Itay Tarush. He is a principal investigator at the Weizmann Institute of Science. He is an internationally recognized leader in single-cell and spatial genomics, especially in the study of human tumors, including gliomas and brain cancers. And Dr. Tarush pioneered the application of single-cell RNA sequencing to solid tissues, revealing how cellular heterogeneity can drive disease behavior. Our third panelist, Dr. Jing Lu, is Party Committee Secretary at the first affiliated hospital of Dalian Medical University. She is the director of the Key Laboratory for Regenerative Medicine and Biotherapy, where she has been instrumental in establishing China's first comprehensive stem cell clinical research platform. She also led the country's first registered clinical study for stem cell treatment of cerebral palsy. So we have three distinguished panelists joining us today. And my first question to you all is, what in your view is the most transformative breakthroughs in neuroscience or life sciences that have such profound impact to the society and also for all of us to consider? So maybe I start with you.
Thank you, Professor Ip. It's a great honor to be here. So I personally come from the clinical and AI world. So I'll let you imagine what my answer to the question, what excites me at the moment is. So definitely AI, but let me clarify what I mean. AI, the hype that we have at the moment of AI is essentially due to the fact that for the first time we can make sense of the semantics of stuff like text and images. But that's not what excites me about AI in research. The way I see AI is a sort of shift in the traditional Galilean method of science, which was originally envisioned as the good science makes the observations, drives some hypotheses and checks them experimentally. Now, this is all fair and good as long as you are in the mesoscale where you can make observations. But once you go down into the microscale, you encounter a problem that you cannot directly observe the phenomena that you want to make hypotheses on. So what can you do? Well, that's when omics can be an answer. You start without an hypothesis. You just collect a bunch of data, ideally a big database, and then you use AI in the sense of machine learning to make some observations. It's like your pair of eyes in that huge amount of data, and then you drive some hypotheses. Now, I was honored during my PhD, I was working for most of my PhD in the computer lab in the University of Cambridge. So I was the only biomedical person in an ocean of computer scientists. Mm-hmm. It was a tremendous mindset contamination experience. I'll tell you more later on, but that's what really, the way I think AI can be extremely impactful in today's research.
No, I totally agree. I think AI can reveal a lot of hidden patterns, among vast, database and all that. So, and also I do feel strongly that collaborations across disciplines is the way to go. So I'm glad to hear that you were immersed in that environment and was able to have all these tremendous achievements. Yeah. Maybe Itai?
Yes, thank you. So I want to continue to talk about a lot of data omics, but specifically in the context of single cells and our ability to not just take an entire biological sample and treat it as one entity, but break it down into the single cell and then produce these omics, very rich data sets for one cell at a time. And this is basically coming from just gradual improvements in the sensitivity of profiling for RNA, DNA, proteins, and other molecules. So once the sensitivity became good enough, we started to apply these methods for individual cells. And I think that led to a big transition in how we think about the complexity of the tissues. Of course, the concepts of diversity and complex structure of tissues was not new at all. But when we were not measuring it so much, at least not at that depth, it was much more difficult to think about many aspects of this diversity. So I think the ability to profile many samples at high resolution and get a lot of information for every individual cell is really changing many fields and how we address many diseases, in particular cancer, which is what I'm studying.
Yeah, I also totally agree. I think, the advancements in single cell and spatial omics really allow us to profile the brain, at an unprecedented resolution. Also, the way that we can really identify the specific cell types that might be, that may play an important role, under pathological conditions. I think this is something that we were not able to do before. So the combination of all these technological breakthroughs are truly invaluable. So, Dr. Liu?
Okay. Good morning, everyone. Thank you for Nancy's introduction. As a neurologist, I have been closely following the fundamental question whether neurological disease can truly be cured. Considering the central nervous system's limited capacity of self-repair, our research has shifted to the therapeutic potential of stem cell therapy. We are currently focusing on three promising strategies: neural stem cells, umbilical cord-derived mesenchymal stem cells, and stem cell-derived extracellular vesicles. These therapeutics promote neural repair through cell replacement or immune modulation or other, and bring hope to patients with neurological disorders. In China, a dual registration system for both institutions and projects was introduced. In October 2016, the China National Health Commission approved 30 stem cell clinical research institutions, including our institute. In January 2017, we launched China's first project, stem cell treatment for children with cerebral palsy. The one, neural stem cells, represent a leading candidate for the treatment of central nervous system disorder. We developed a dedicated neural stem cells prep preparation technologies and validated its therapeutic potential to cerebral palsy in preclinical studies. Notably, we have adopted a patch-based intranasal delivery method for the administration of neural stem cells. This approach significantly enhanced the migration efficiency of stem cells to neural injury sites while improving safety and patient compliance. The intranasal delivery bypasses blood-brain barrier in a non-invasive manner, reducing risks and increasing treatment acceptability. Clinically, in a phase 1 and 2 trial involving children with cerebral palsy, we observed safety and promising signs of efficacy. Participants showed immeasurable improvements in gross motor function and muscle tone regulation and cognitive responsiveness after transplantation. I think this is supporting the feasibility of this approach. And the second, umbilical cord derived mesenchymal stem cells exhibit strong immunomodulatory and neuroprotective properties. Our team manufactured a clinical grade MSC, which has passed the authoritative review and inspection by China National Institute for Food and Drug Control. Their biological properties and their microbiological and biological safety and efficacy meet the highest standards for clinical application.
That's very exciting.
It's very difficult.
Yes, I'm sure. So may I invite our 5 laureates in the audience to maybe also comment on your thoughts on, in the last 5 years, for instance, what do you think are the most transformative breakthroughs in neuroscience and life science that are so impactful and make such a big difference in our research and also in potential clinical applications? Please.
Well, the one thing I would say, and I'm hopeful we have some young people in the room who are thinking about careers in science. I worry a little bit when we talk so much about exciting technologies that becomes the focus. One of the many powerful and useful things I learned from my mentor was good science starts with the question you're asking and developing a taste and instincts for a good scientific question. And then you can have a discussion about what technologies to use. But I fear what I've seen happening is we have so many really exciting, powerful technologies that young people come to me and they say, I want to do spatial omics. And I go, well, what's the question? And they don't know what the question is. They just want to be the next person. So I would just caution the young people, I think it starts with the question. And secondly, I think there's a lot of complementarity between what we used to call reductionist biology and what used to be called systems biology. Maybe it's been rebranded. But, I think, follow what is giving you joy. And I like science that involves puzzle solving and hopefully being a bit clever. And I like getting into mechanisms. So for me, I do what I try to do is reductionist biology, and then I complement myself with smart people who do systems biology. But again, start with the question and find what gives you joy, and you're on a good path.
Yes, I'm in full agreement, Professor Kevin Jr., because I also feel that, doing science is our passion, right? And I think for us to have, interest in doing basic science is definitely the way to go, especially for our junior colleagues. In parallel, though, when there are advancements in technologies, and if we can utilize them, or if we can leverage these technologies to do work that we previously could not do, like the single-cell spatial omics, I think is a very good example. So I don't think the two are mutually exclusive. I do think that they actually enhance each other and they can synergize. So I'm totally in agreement with you, and thanks so much for bringing that up, because for the students or researchers young researchers in the audience, this is something we must keep in mind. Yeah. Other comments? Maybe audience can also ask questions about the laureates' talks, if you have any questions. I can't see. Yes?
Thank you for your wonderful panel discussion. A lot of you talked about the huge potential for translation, but there are lots of barriers to adoption in clinical practice. And I'm wondering if you could talk a little bit about the pathway to true adoption in the clinic from your perspective. Thank you.
That's a wonderful question. That was my question too. So, I mean, again, question for the panelists. What do you think are the greatest opportunities as well as the challenges when you want to translate your scientific discoveries into clinical practice? Because after all, I think this is a continuum, right? As scientists, we have, scientific discoveries. Sometimes the discoveries will stay in the lab. We publish papers and they don't go beyond the lab, but to take our scientific discoveries, from the bench to bedside, I think, again, this will require a lot of collaboration with different key stakeholders. And again, like you, Julio, being so successful in translating your discoveries, I think you might want to share, be the first one to share.
Thank you. I would like to start by commenting about
Sure.
the scientific questions comment that has just
Sure.
been brought up. I couldn't agree more.
Yeah.
I like to say that it's a bit of a exaggeration, but during my PhD I spent 3.5 years looking for a question, half a year creating a solution for that. It's an exaggeration, but there's some truth in it. And what I could experience being surrounded by people that had the knowledge of the tool, the computer scientists, but not necessarily the domain, the problem, is that on average, my computer scientist colleagues were extremely clever people, and I had a ton to learn from them. When they applied their cleverness on medicine, often they created brilliant solutions to known problems. And I can list a number of examples. Just to give you one, a brilliant colleague of mine, extremely clever, created a very advanced GNN architecture that was able to tell you the side effects of a drug based on the molecule. It was brilliant. However, it was trained on the publicly available data sets, which tell you only if there is or there isn't a side effect. But the sampling of the current pharmaceuticals is very diverse. Some drugs have been taken by billions of people, some by dozens of people. So if you tell me that a certain drug has a side effect, is it in the sense of 1 in a million or 1 in 10 people that take the drug? If you don't tell me, clinically, that architecture is pointless. So I think that the greatest challenge that we are facing as a scientific community is that going forward we will need more and more people that have a dual background. On one hand, one toolbox, which often must come from the engineering world, and on the other hand, a domain kind of background. Otherwise, you risk to solving non-problems.
Yeah, but that can be achieved perhaps through collaboration, right?
Oh, absolutely.
Yeah.
But at the same time, you need to at least have the mindset
Sure.
of the two kinds of souls that you want to interpret.
Correct. No, I'm again in agreement. I do think that, the only way you collaborate across disciplines is you have an open mind, because it's almost like, learning a new language. Because when I collaborate with my colleagues in engineering, it really takes a long time because we speak different language, but as long as we put in the efforts, then it will work out beautifully.
Totally.
So I'm in agreement. So the biggest challenge you see in the translation pathway is to collaborate with others who are open-minded and have to share goals and be willing to work together. That would be the key, right?
Yeah, absolutely.
Yeah.
But also to ensure that the young scientists more and more have this dual kind of background. That's
Right,
what I mean.
in terms of the training, in terms of education.
Exactly.
So again, I think for universities, we have a very important role to play. Because the programs that we offer our students, should take that into consideration, to have programs that will allow the student to learn not only about, their major, but also maybe emerging areas of interest.
Because if you have a limited toolbox, you will say you have only the 4 arithmetic operations. Probably someone has already tried that to solve your question with the toolbox. If you have a big toolbox but you cannot identify meaningful questions, you're making brilliant solutions to non-problems. So you need to have a sense of both.
No, I totally agree. So it's the mindset of the graduates, right? That they have that kind of openness.
There was a brilliant paper a few years ago that was published on Cell, an editorial paper. It was, "Can a biologist fix a radio?" It's fascinating. I suggest you to read it. It tells about what happens if you give a broken radio to an engineer or a biologist. And the engineer would make schematics of the circuits and eventually would be able to repair the radio, the broken radio. The biologist would, it's really hilarious. So if look at the model of the radio, order a bunch of radios of the same model, and try to replicate the phenotype by removing individual parts. And long story short, it's hilarious. So it would start naming the antenna, the battery, stuff like super important component, ultra super important component, stuff like that, as we do with genes. Eventually, they would not be able to repair the radio unless the phenotype is only one broken system. If that's one broken system, then because they can replicate the phenotype. But if it's more than 2 missing parts, they would not, because the language of the biologist does not fit the language that is best to describe the problem. And my question to you is, what would a biologist with ChatGPT would he or she be able to repair the radio? I think that's an interesting question.
Yeah, maybe the laureates can respond to this. Okay.
Sorry, I had a follow-up question, but I believe the floor is open for response from one of the speakers.
Yes, sure.
Oh, okay. My follow-up question is, what kind of projects or what kind of science do we fund going forward? I'm a molecular biologist. I was trained in the reductionist approach. I recruit PhD students now. Do I want to continue training them to go after one gene, one protein, transcription factors? That's what we do. Or should we push them into being really disease-focused? Should it be stem cells, a clinical application that will actually bridge across and have not translational, but a clinical impact? And if it's a split between the two, for a country like UAE, which is where we're based, is it a 50-50 split? Or is it majorly translational clinical work that can have real impact in the next 5 years? Or is it pure science for discovery for the sake of science?
Well, it's a very interesting question. I don't think we are able to address the funding mechanisms in the UAE, but I can share with you that actually there are funding mechanisms for both types of research because they are both important. I don't think we should focus just on the translational part because without basic research, without innovation, it would be very hard to make progress. So I think both are important. I suppose for this particular panel, let's focus our discussion on the topic of, how to, just now the question was, what are the challenges that we should overcome to take our scientific discoveries to, to clinical practice, for instance. So maybe, Itai, you have some thoughts on this?
Yes, so one of the more recent advances in the single-cell field and the spatial field is that we can begin to apply these methods not just to fresh samples, which makes it very complicated logistically to work with patient samples, but also to archival samples, for example, to formalin-fixed paraffin-embedded samples. And this now opens the door to analyze more closely the clinical samples, because we have that from standard clinical practice. And I think this is one example of a more general effect, that the ability to produce so much data and so rich information that allows us to address many questions directly from just one patient sample has caused a shift from mostly focusing just on model systems to also analyzing more and more directly the human patient samples. So this larger shift from model systems to human samples makes it easier to now study the real disease at its real context. And it also highlights the limitations of the model systems and also fueling a drive to have better model systems. So I think this is, in general, improving the connection between the clinical world and the research world, and allowing us to do even some basic research directly on patient samples. So it's not directly addressing the question of translating it, but at least it makes it easier to make the initial discoveries and the models closer to final implementation.
So it increased the success rate, for instance, right? I mean, especially for drug discovery efforts. If you have human model systems, then perhaps it will be, a bit more, with the success rate a bit higher, maybe. Yeah, maybe Dr. Liu, you have any comments on this?
I answer this question from clinical doctor. Okay, if we want to translate therapeutics from preclinical validation to clinical reality, an institution in hospital, we need to do as bylaw. One, we must build an institution that is meet the GMP standards and approved by the national, maybe China National Health Commission approved. Okay, this is one. The second, we need to develop the standardized scalable technology for stem cell or vesicles isolation, purification, and industrial scale production. And this must pass the authoritative review and inspection by the national China National Institute for Food and Drug Control. Okay, this is second. And the third, we need to approve the certification for who and what kind of cells for what disease. So, about my project, that is neural stem cell treatment for the children with cerebral palsy. That's by Dr. Jing Liu. That is okay enough. So back to the question. Can neurological disease be cured? I think a cure may not be a single injection that runs a disease overnight. Maybe it may be a regenerative intervention that has the degeneration and restore meaningful function, altering the disease trajectory and giving patients years of quality life back. Okay, that's my answer.
Thank you, Dr. Liu. Maybe I also would like to invite any of the 5 laureates, if you want to comment on this, the challenges in translating scientific discoveries to clinical practice, please.
I don't want to take time from my fellow laureates since I've already spoken, but you know, my heart went out to the young lady who asked the question about translation, and this is about translation. So I've worked with many pharmaceutical companies, biotech companies. They all say the same thing, and that is, if you in academia want to do applied science or translation, that's wonderful, but we're actually pretty good at it.
Yes.
But what we can't do, for a variety of reasons, is the kind of basic fundamental research that we've always relied on the academic sector to do for us. So I fear that young people today are under too much pressure to justify what they're doing in terms of its translatability, its applications. I realize my last slide was about the translational aspects of my work, but what we really tried to do was solve what I thought was an interesting puzzle. And in the back of my mind, I thought, you know, if we generate enough knowledge of the system, maybe there will be translational opportunities. But if someone had put a gun to my head and said, we're only going to fund you if you have a new treatment for kidney cancer in the next 5 to 10 years, I would have found another line of work. So I think wise governments, wise funders would acknowledge this is how really progress happens. Another way to think about it is companies largely do engineering, which is using rules to do useful things. They rely on academics to do science, which is learning the rules of the system. And to me, that's where academia, I think, really shines.
Yes.
And so the last thing I would say to the questioners, you know, I don't think you want to turn your lab into a vocational school just training technicians for companies. I think you want to hopefully generate those next creative people who are going to completely change the way pharma and biotech think about a problem.
Well said, yes.
I just want to pretty much echo, because I feel very strongly about this. I think translational is great. As I mentioned, we're doing this as well. But I think this curiosity-driven research is kind of taking a backseat even in academia, including our own American National Institutes of Health. And I think this is a big mistake. Even, you know, pharmaceutical companies, if you ask which compound became a drug for what indication, most of them start doing something else. And this is just one evidence to say that we cannot predict where the cures are going to come. We cannot predict what indications they're going to be good for. And of course, CRISPR-Cas9 is a great example of this. One could have said, who cares about how bacteria defend themselves from viruses? We should never spend money on this. And so whenever we have a finding and some journalist asks me, how's this going to help humans? And I, say how this could be great next pain target, for example. But I always like to emphasize, because I think everybody needs to know this, is that curiosity-driven research needs to be supported. Without that, nothing else will work. And I think it's very important to emphasize this to students, to government agencies, etc.
Absolutely. Well said. Yeah. I do think that all of us have the responsibility to remind our government that, on one hand, of course, they want, short-term return, etc. But I do think that the emphasis on supporting fundamental science is so important. And all of us in the academia will always keep this in mind. So we have internal funding to support basic research, and we also urge the government to have a balanced funding mechanism, not just always asking for impact, because it's very hard to predict our fundamental research when it would have impact. So the lady who asked the question earlier, about the funding allocation in UAE, I would say that let's emphasize to them that supporting curiosity-driven research is very important, and collectively we should, voice that opinion from the academic arena. So again, one way to promote collaboration with industry, in our experience, is to form joint laboratories so that the academics and the industrialists, will have a shared goal, so that we can work together and the fundamental research discoveries from universities can have a way, can have the opportunity to be translated by working in partnership with industry. So I noticed that my time is up. So again, we had very good discussion, and again, the emphasis on basic research, the leveraging the opportunity of technological advancements, how we can perhaps push the translation path by working together with industry. I think it comes out very clear. So again, I want to thank my 3 distinguished panelists. I want to thank you all for being here, and I am going to also remind you that we will now take a 10-minute tea break, and please return for the next session that will be on genetic science. That will happen after the tea break. Thank you all, and I would like to invite our laureates on the stage. Thank you.